this is Beren. We're going to talk about patient on a chip. We are a multi-organ chip company with 2, 3, and 4 organ chips that is available, which can use different cell sources to make a fit-for-purpose assay with the organs of interest. Our whole effort is to solve the problem of translational research, which has high failure rates. For a new compound entity, animal models are used as a systemic solution, which is not human, or human cell culture solutions, which are not systemic. In the end, with the high failure rates, makes a new compound entity reach to the market very expensive. To have a systemic and human solution, we established numerous assays for safety and efficacy. As an example of our recent projects, I can show you our functional coupling of human pancreatic islets and liver spheroids on a chip towards the type 2 diabetes model. And we kept these tissues live for 15 days on the chip. And only under the co-culture conditions, liver and pancreatic islets showed glucose uptake and insulin release in physiological amounts. And when the islets were cultured alone on the chip, they actually reduced their insulin release over time, losing their functionality. And in the case of human skin and tumor co-culture, we used lung tumor cell line and EGF receptor antibody as a proof of concept study. And with this, we showed the efficacy of the treatment in the chip, that the tumor cells showed cell death and expression of apoptotic markers, while the skin showed the known side effects, seizing their proliferating cells, and basically both showing the safety and efficacy study on the co-culture. And coming to the higher complexity level of the ADME platform, we used 4-organ chip, which has a blood circuit and a kidney circuit with the interface of kidney cells on a porous membrane. And this chip is PBPK compliant to show the administration route, the metabolism with the liver, neuron as the target organ, and kidney for the excretion. And we could keep these cells alive for 14 days. And coming to the human on a chip concept, this has to be from a single iPSC donor where we bank the different donors. And we already have established more than 10 organs from the same donor. And we differentiated them and already testing them on the human on a chip platform to have a universal platform in the highest complexity. And how to mimic the patient's body on the chip? Of course, we have to start with the donor from the cell donation from various different organs with different genetic backgrounds to have a variety of stem cell bank and differentiation of the organ equivalents from these donors, and to have the human body on a chip with the same organs with the genetic background. We can culture them in longer time on the chip and eventually induce the disease on the same healthy patient to be able to treat this patient on the chip before even the patient was sick, to be able to test the new compound entities eventually that would be used for this patient. Thank you for listening.